I've always been very interested in emergence, so it's great to see that there's a Dutch Emergence Institute. So I thought I would talk today to you about, I think, an interesting example of emergence. I'm going to claim this, that simplicity naturally emerges from the algorithmic way of thinking about the world. And to begin, I want to tell you a story. I introduce you to a story, really, called Borges' Library, by the great Argentine writer, Ard Louis. And in this little story, he talks about a library where every book, this library is made of 410 pages, 40 lines, each 80 characters, 22 letters of the alphabet, and a few pieces of punctuation. And the interesting point is that every single possible book with these characters is in this library. So in the story, at some point, the librarian realizes something amazing. The library is total, perfect, complete, and whole. That is, all that is able to be expressed in every language, all the detailed history of the future, the autobiographies of the archangels, the faithful catalog of the library, thousands and thousands of false catalogs, the proof of the falsity of those false catalogs, a proof of the falsity of the true catalog, the Gnostic Gospel of Basilides, the commentary upon that gospel, the commentary on that gospel, the true story of your death, the translation of every book into every language, etc. It's really fun to think about this. So in that library, there exists a book that tells you the purpose of your life. And so in the story, people start looking through the library, desperately trying to find this kind of their vindication. And of course, as Borges points out, not only is that story there, but some treacherous variation on that story is in there. There's one that says basically the purpose of your life is to stand up right now and kill everybody in this room, etc. And many, many treacherous variations. And the thing is, you may never know when you find the book which one is true. It's kind of interesting to think about this. Every possible story that you can fit into 410 pages that you could imagine is in there. Amazing proofs are in there. You know, if you can unify quantum mechanics and gravity in 410 pages, it's there. But also many things that are subtly wrong are also there. So it's really fascinating to think about. The question I want to ask you to think about today is, every book is equally likely because there's very large numbers of books, 10 to the 1.8 million, I calculate number of books, but every book is equally likely. But is every story equally likely, right? Now, there are stories that are quite complicated, like maybe the detailed story of your life up to now really needs at least 410 pages to be told. But there are very many simple stories, Little Red Riding Hood, etc., that could be told in many different ways, and so might appear many times in this library. So the instinct I want to give you is that every book is equally likely, but every story is not probably equally likely. Some stories are going to appear there more often than others. So I'm going to try to play with this intuition a little bit and think about this more abstractly as a possibility space. So physicists like to think about state spaces, right? This is a possibility space. Here the space is not all sets of positions of momenta, but the space is all possible stories. And the question is, what can we say about those kind of possibility spaces? I'm going to introduce to you a field of theoretical mathematics or computer science called algorithmic information theory. How many of you are aware of algorithmic information theory? Does that say something to you? Okay, a few of you are. So I will skip over some subtleties, okay? There's some great people here in Amsterdam who have written one of the great textbooks on algorithmic information theory. I'll skip over the details for the experts, but I'll hope to explain it to you in ways that are clear for everyone else. So I want to introduce this with two infinite monkey theorems. So infinite monkey theorem, you see it throughout science and throughout pop culture. It even made it into The Simpsons and into Richard Dawkins' books. And the idea is basically a monkey types on a typewriter, right? So he randomly types, or for those of you who are a bit too young to remember typewriters, into a word processor, right? So everything, what you type is what comes out, right? So how likely is a monkey to type something like me thinks is a weasel, right? And a typewriter, 26 letters plus a space, it'll take 1 over 27 to the power 28. To take on average something like 27 to the 28 keystrokes, it'll take an extremely long time. And for the monkey to type anything longer that's interesting, it'll take even longer. But what if I do a twist on this and the monkey is typing into a computer that interprets code, right? Doesn't really matter which language it is, some language, right? Then if you look at the outputs of that monkey typing, right, you might notice certain things. You might see that a string that's quite long, 0101 repeated, will appear much more frequently than you'd expect it to appear if a monkey's typing on a typewriter. And the reason for that is because there exists a short code to generate that sequence. So if the monkey's typing into something that's coding things, then the frequency with which outputs appear is linked to the frequency with which those codes appear. For every string, there is a very simple code, which is print that string, right? So there is a code that says print 01 50 times, and that is print 010101. That's a long description. It's not very much different from the monkey on a typewriter because you have to put print in front of everything. But there are also some strings for which there are shorter description. And already I've gone to strings. So for those of you that are used to computer science, everything can be reduced to binary strings typically. So I'll look at binary strings and assume that the things I say about binary strings are true for patterns in general. So we're used to in physics thinking about Shannon information, Shannon entropy, which is basically a measure of surprise. So if I have, say, a coin that I can heads or tails 0 or 1, then I say if I have a low entropy that always give me one output, then it's all zeros, or high entropy, which is, as far as I can know, a random sequence of zeros and ones. If I just have zeros and ones, I may also have a sequence of 0, 1, 0, 1 repeating, which is still high entropy sequence, equal number of ones and zeros. But I might look at that and think, well, there's a difference between these first and the third sequence and the middle sequence. Can I capture that difference somehow? And to introduce the fundamental way of thinking about this, I need to take a step back and think about computation in general. So this is the universal Turing machine. It's a little picture of it that Alan Turing famously worked out when he was just about 23 years old. It's pretty amazing. It's a very abstract computing device. And one of the reasons for that is because it turns out that a lot of theories of computation don't really depend on which computer you use. They're just properties of computation. So it doesn't really matter what it's like. This is a computer that can do any calculation that can be calculated. What's kind of interesting is that Turing came up with this machine not to found the theory of computation, which he did, but to prove that there are things that computers cannot calculate. And this is linked to Gödel's undecidability problem. And so one of the interesting things is that in his paper, he proved that he proved basically that there is no algorithm that will tell you whether a certain code that you give will always halt and give you output or just run forever. It's called the halting problem. It's a very deep fundamental connection. A bit later, people started thinking about universal Turing machines and realizing that the question I gave you about sequences can be answered more formally in terms of monkeys on typewriters, monkeys on keyboards, monkeys on computers. And Kolmogorov and Chaitin basically worked out that the fundamental description of the Kolmogorov complexity, it's what's called K, description of a string, is the length of the shortest program that will generate that string on a universal Turing machine. And since the universal Turing machine, if you have a particular one, can always can do any calculation, it can always include a compiler to any other computer's universal Turing machines. It doesn't really depend which universal Turing machine you use up to compiler terms. And what's interesting is it basically tells you this string has a short code, print 0150 times. This string, to my knowledge, has no shorter code than print the number. It's a complex code. And one of the things you should be careful about is that the string I show you right here looks pretty random. In fact, it satisfies it's Borel randomness test, a randomness test, but it's actually the binary version of pi, which does have a short code. So it's hard to know from a string necessarily whether you found the shortest code. Okay. But this is a formal way of defining the complexity of a string. And it has many nice other properties. So for example, if I want to ask what's a random string is a string for which the Kolmogorov complexity is the length of the string. Basically means there is no shorter code than print the string, right? Then it's random. It has no pattern. Right? There are also other interesting properties like the Kolmogorov complexity of a set can be a lot less than the elements of the set. So the Kolmogorov complexity of Borges' library is that short description of 410 books. That's all. That describes the entire library. Of course, in that library are incredibly complex books that take a lot more information to describe. Another interesting thing that you can kind of work out is that the vast majority of strings are complex. I'll give you a really simple counting argument. So let's say I've got some magic compressor, a zip or something, and I want to compress strings of length 10. Okay, so strings like 1, 2, 3. So there are 1,024 strings of length 10. Okay. If I want to compress them, I can compress them. I have to have a short description to compress them. If I add up all these strings together, okay, and only 1,022. So there's two strings that can't be compressed. Half of my strings can be compressed by just 1 bit, right? 3/4 by at most 2 bits, right? And the vast majority of strings can't be compressed. Roughly speaking, 1 over the number of strings I can compress by p bits is 1/2 to the power, or the fraction is 1/2 to the power p. This tells me in Borges's library, the vast majority of books are just gibberish, right? They can't be compressed in any kind of way. When you take a book on your computer and you compress it, you can often change its information volume by a factor of 2 easily. If you think about how many bits you've compressed that's extremely high. If you think of the fraction of all possible books of that length that you can compress by that's 1/2 to the power of the number of bits that you've compressed it by. It's hyper astronomically large. So this tells us that only a tiny, tiny, tiny fraction of all books in Borges' library would be what we would recognize as stories. Okay, that's one thing to remember. And the vast majority of strings are complex. So if I just take strings at random, okay, and I've got some compressor that gives me a complexity measure, which is the shortest code that can describe that string, then I know that my probability over upon picking random strings, as much as complexity is going to look something like this. If I do a log of it, it's going to go like that, because it's 2 to the power k. And this is k is equal to the length in my perfect compressor. This is k is equal to length n minus 1, and 50% of all strings will be in that first little bucket. I'll come back to that in a minute. You've got something to remember about strings. So this is kind of interesting. Yes, go ahead.
So if I think about it in this way, like at some point, so there's only a very few books in the library that have small complexity, but the ones with the smallest are actually not interesting.
No,
because they will just be a string of A's or the whole way through. So how can you find the interesting ones? What in terms of this complexity? Because you want to, I guess, the concordance will be a bit more.
Yeah, I'll come back to this in a minute. You'll see that basically. So one of the complaints of complexity is that basically, 0 is uninteresting. It's all the same.
Yeah.
This is just random noise.
Yeah.
And so somewhere in the middle there is the interesting thing. And if you look at the history of complexity theory, there's been huge amounts of discussions about, what kind of complexity measure should we use that would pick that up, right? It's actually an unsolved problem, even in simpler physical problems. Like for individual systems, you can find complexity measures that tell you something. But our intuition is the interesting sort of complexity science, if you want to, was very popular in the 90s and early 2000s, is trying to find measures that pick that up. And there's no agreed upon measure that does that because it's a complex. I think but it is true that most interesting stuff happens in the middle. And you'll see I'm going to show you some examples of exactly that a bit later on.
How long does it take to compute it, depending on the length of the string?
Compute what? So this depends on the measure that you use, right? So here I'm assuming there's a magic compressor that can compress everything perfectly, right? But if I want to, I can also tell you that formally it's uncomputable. Okay? So it takes you longer than anything you can imagine because it's uncomputable. In practice, what I'm going to show you is I'm going to use a compressor, right? And those could be super fast or super slow, depending on how nice you want to do them. And I'll tell you a bit why I would do that. The argument would be, well, if I can compress a string with a compressor, then I know it's got a simple description, right? There may be incompressible strings like pi that are nevertheless short.
Can I think of this in the same way as like the work on the cellular automata? So Lindgren's measure of complexity.
So there's links between those, right? There's many links between all kinds of measures of complexity. Right?
Are these books like procedurally generated? This is the idea.
Yes, right. So the point is what fundamentally, if you have a universal Turing machine and some cellular automata are universal, right, then you could use cellular automata to generate them, and it will be the same because you can always write a compiler between Fortran, which is also universal, and your cellular automaton. So what is the interesting thing about this story is that the idea that the shortest description of string is somehow special did not come from Kolmogorov, actually. It came from a very eccentric American computer scientist called Ray Solomanoff, who he was actually very influenced by a philosopher called Carnap and was interested in formalizing the problem of induction. And he basically said, what if I try the following problem? I want to generate a particular output, okay, a particular string. And I'm going to do that by just randomly feeding programs into my universal Turing machine. That's exactly the monkey on a typewriter, on a computer, sorry, example I give. So I've got a universal Turing machine. I'm going to simplify my life is my language is binary, okay? So how likely do I produce a certain output X, like pi, for example, right? Where it's a sum over all programs that generate X, right? With each program, how likely am I to make it? Well, if it's binary, the probability I get a particular program is 1/2 to the power of the length of that program. So I sum over all those programs, right? What's the first and largest part of that sequence is going to be the shortest code, right? That's going to be the most likely one I'm going to find of all the possible lengths. But there may be many more that are longer, right? So this is where the Kolmogorov complexity actually comes in. And this is what we call this object here, algorithmic probability. What's the probability I generate something on random sampling of strings? And I like this because I like this quote from Marvin Minsky, one of the founders of AI, who said not long before he died, it seems to me the most important discovery since Gödel was discovered by Chaitin, Solomanoff, and Kolmogorov of the concept called algorithmic probability. Everybody should learn all about that and spend their lives working on it. And so I'm going to hopefully convince you that physicists should also spend their lives working on it. It's just, so that's the idea there. There's many problems with this. One of this is that the probability is really not probability, but a semi-measure. There's lots of interesting things about what it does. I'm going to skip that for now. This is because some of the programs you have to wait forever to know whether they'll halt or not, so you never really know for sure. Okay, but it's a really beautiful set of ideas, and there's many grand claims for this universal distribution. Claims that it solves problems of priors, claims that it supplies the justification of outcomes razor, claims that it will generate undergird general AI. That's not quite. It turns out if you look very carefully, that's not quite right. There's a very good philosopher who used to be in Amsterdam, Tom Sterkenberg, who's now in Munich, who's worked out subtly why these things don't quite work. But there's lots of interesting claims about this particular algorithmic probability itself. What's super interesting, and this is a theorem that I think everybody should know, is that actually 50 years now, Leon 11, also known for P versus NP, if you follow computer science, proved that not only was this largest term a lower bound because there's at least one code that generates it, it's also up to order 1 term, so terms that are independent of the output, the upper bound. So this is a tight bound. In other words, it tells you that the intuition, if you have a simple, it formalizes the intuition that I was giving you already, which is that if I have a simple output, so that means an output I can generate by some short code, then if I randomly type into it, I'm going to get it with high probability. That's basically what it's formalizing. You might ask yourself, why have you not been taught this as an undergraduate? This is a very profound fundamental theorem. It basically tells you that you should assume that simple things are exponentially more likely to appear than complex things as a kind of null model, right? And the reason you haven't been taught that probably is because, well, this only works for universal Turing machines. Most of the things in nature we care about are not universal, although some are. The complexity itself of here is uncomputable formally. And then this is order 1 term, which is uncontrolled and hard to know exactly what it is, right? And if it's large, then it'll dominate and you don't really know what it is. So it's been, and also sociologically, the kinds of mathematicians who are interested in these problems are typically people interested in foundations of mathematics, where you need to be extremely careful about every single assumption that you make, or things go wrong, and they're less interested in applications. But for physicists, that has never held us back. Okay, and so when I first learned about this theorem, I thought that's amazing. Okay, I'm sure that if I go away from a universal Turing machine and I loosen some of the assumptions, my exponential preference for simplicity shouldn't just suddenly disappear. That would be very odd. And in fact, you can formalize this a bit more. So in theories of computer science, you talk about universal Turing machines, which can be mapped onto a certain type of grammar. So Noam Chomsky, very famous linguist and political activist, wrote his most famous mathematical paper by saying there's actually a hierarchy of different machines that can do different things, that can calculate different kinds of grammars. and universal Turing machine at the top. And then there's other ones like context sensitive, context free, regular ones, which also include Markov chains. And the question you can ask yourself is, well, if I look going further down this Chomsky hierarchy, okay, will I still see this universal Turing machine? Turns out that it's super hard, and we're working on it, not able to prove it for any of these other levels. In fact, to prove the coding theorem, you need undecidability. It's just a bit unfortunate because these things are decidable. But what we did do is a typical physicist approach. So it's actually two PhDs long, my students Kamal and Chico. And what we basically said is if you have an input-output map of some kind, so you have inputs and they generate outputs, there are more inputs and outputs, I can put a probability on the probability I get a certain output. Then as long as the map is relatively simple, that means that the map doesn't take a lot of information to describe, then it doesn't grow with system size, for example, then I can at least prove an upper bound, which says that the probability I get a certain output is upper bounded by a half to the power of Kolmogorov complexity. Now that I can't calculate. So now I've got a little squiggle above my K where I say I'm going to approximate my Kolmogorov complexity by some compressor. And we're going to use, for example, Ziv 76, which is one of the compressors, which we think is a good one. Okay. But we're going to add a prefactor because we don't know whether it scales properly with the true Kolmogorov complexity, and add an offset which tries to pick up the order 1 terms. Okay, so it turns out that if you apply this to you can apply this to a wide range of different systems. Here we have a number of systems. I'm going to discuss this one in a minute. This one here is a circadian rhythm model. It's coupled differential equations with a bunch of different parameters. You can look at the output of that model, and you can ask yourself, if I just randomly change those parameters, how complex, what happens to the output? You can describe this, you can discretize the output, right, as a string, ask yourself, do I get complex outputs or simple outputs? And it turns out that you're exponentially more likely to get simple outputs than complex outputs. These are L systems. So these are little, actually from a Hungarian-Dutch biologist called Aristide Lindenmayer, who used to be in Utrecht. And it's used for looking at plant growth. And you can show if you just randomly make codes for plant growth, that simple ones are exponentially more likely to appear than complex ones, etc. So the question is, we call this simplicity bias, it's a bias for simplicity, and can we see this in systems that we actually care about in nature? Does that make sense? So that's the idea. So this is beautiful, elegant coding theorem by Levin, right? That's hard to apply in practice. And so we're going to just do a physicist approach where we're going to try to apply it anyway and see what we see. Yeah. So I'm going to look at biological possibility spaces now, rather than possibility spaces stories. So you may know that proteins are made of strings of amino acids, size 20, RNA, which can also be functional, so they can do catalysis or structure. They got 4 nucleotides. So the number of possible, those libraries get very, very large. If you made every protein of length 37, which is really a peptide, it would weigh more than the Earth. Okay, if you made everyone of length 58, it would be more than the observable universe. For RNA, that's 126 would be more than the observable universe. Still, these are really short sequences. The main length of proteins in your body, at least according to Wikipedia, is 476. If I made every protein of length 476, it would weigh 10 to the 500 times the mass of the observable universe, which is unbelievably and unimaginably large. So these possibility spaces are unsearchably large. Nature has only searched a tiny fraction of those spaces. And it's interesting to ask yourself, what do those spaces look like? How come we can find function in those spaces? Lots of interesting questions appear from this possibility space arguments. But I want to just remind you that for 55, I'm going to talk about in a minute, every possible strand weighs 10 to 10 kilos. I got interested in this because actually a friend of mine, Simon Conway Morris in Cambridge, pointed out that this experiment by Jack Szostak is a famous Nobel Prize winner, lots of beautiful work. And one of the things he's helped pioneer is in vitro evolution experiments. So you do experiments in a test tube. In this case, you're taking RNA strands that are functional. He looked for a particular function of self-cleaning enzymatic activity. You can put a bunch of them in a test tube. You can run it through an assay, pick out the ones that show some function for the assay you're interested in, and then if you use a noisy copying mechanism, you take those out and you can repeat that. You can basically make Darwinian evolution in your test tube. And so in this paper that they wrote now 20 years ago, 25 years ago, he and his collaborator pointed out that every time they ran this selection on a particular function, they found the same structure, which it's called the hammerhead ribosome. It looks like this. Okay. So I was very interested in this because I thought, well, is that surprising that you find the same thing every time? Well, if you made every possible sequence of like 55, it weighed 10 to the 10 kilos. I estimate he used about 100th of a gram of material in experiments. That's still a lot of molecules, but it's a very small number compared to the space. And so why does he keep finding the same shape each time? It's a little bit like putting your hand into a haystack and finding a needle every time. Okay, how come that happens, right? It's very surprising. And there's really two possible answers to that, right? One would be natural selection. So this is Charles Darwin's great idea that you've got variations, so offspring are slightly different from yourself, as the noisy copying mechanism does, and then natural selection will pick on some fitness advantage. That's what happens in the experiments. But actually, this is an Amsterdam connection. It's not just natural selection. You have to actually have these variants that appear in the first place. Natural selection can only act on that which it is given to it. And Hugo de Vries, a famous botanist who rediscovered Mendelian genetics, because Mendel's genetics was forgotten for a long time, wrote a famous quote: "Natural selection may explain the survival of the fittest, but it cannot explain the arrival of the fittest." And so the problem here is you're only using 1 in 10 to the 15th of the space of all possible sequences. You can't select on something that doesn't appear. Okay, so there's a fundamental problem here: is why is this appearing in the first place? Selection isn't enough to give you, as far as I can tell, this particular story. So why did I pick RNA? It's because RNA has some very nice advantages. This is a sequence of the 4 nucleotides. G's bind to C, A's bind to U, G sometimes binds to U as well. And this is hammerhead ribosome, this three-dimensional structure. But there's a very simple problem, or a simpler problem, which is the secondary structure. It basically says who binds to whom. So in this sequence, I start from this G, it's got A, you see it, that U there binds to that A, etc. And if I take this sequence, it turns out that the free energy minimum of that sequence is this particular structure. If you look at it, this looks like a hammer. You see the hammerhead? That's why it's called the hammerhead ribosome. And what Shostak and his collaborators did is they put their sequences that they found through one of these folding algorithms and found always this structure. So the question is, why do they keep finding this particular structure? Right? And because it's very fast, I can do really fast calculations on it. I can abstract this out a little bit. So this is a bit of a complex structure to look at. What's kind of cool is if I look at, here's a slightly simple structure for you to understand, I can rewrite this in terms of a notation, which is basically a ternary string. So if I just open a bracket every time I make a bond, and then have a dot when I have no bond, and close a bracket when the bond closes, see, open, 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 dot, 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 close, 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 dot, 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 then I've got a unique description of this structure. And I can ask myself, is this structure simple or complex? I can put it through a compression algorithm and say, is it simple or complex? And then I can ask myself a really simple question: is if I just pick sequences at random, like picking stories, books at random, right? How likely am I to get a particular secondary structure like this? Right? How likely am I to get one of these? And can I say something about that? So our coding theorem predicts that you're exponentially more likely to get outputs that are simple than outputs that are complex. And here we show how that works for RNA. So on the x-axis and the y-axis, I have the probability, the log of the probability that I obtain a certain structure by random sampling. On the x-axis, I have the complexity of those structures measured by how compressible they are. And what you see is that actually the most likely structure to appear, so the structure which has the most sequences as its ground state, is the unfolded structure. That's the simplest structure because it has the sequence for it is just a bunch of dots. That's very simple. And this particular hammerhead ribosome you see here is one of these very high probability structures. The space here has a size of 10 to the 35, so I could never sample that sequence. We estimate there's 10 to the 13 possible structures, so an unbelievably large number of structures. So the ones you're seeing here are a tiny, tiny fraction of all the possibilities. Okay, there are very, very high probability structures. And what I see here is, I'll show you here I've got the probabilities for a number of these structures, and here I've got their complexity, which confusingly for you I called C here and K circle there, I just noticed. But like this is a simple structure, the highest probability one. Here are some more complex structures that are much less compressible and they're less likely to happen. So what's nice about this is I can tell you which RNA structures are most likely to appear without having to know anything about the physics of how they fold. Go ahead.
I have a very simple question. Do you assume that all sequences are equally likely, or do you
Yes,
have a question? I mean, there is of course the notion of favorable interaction, right? Like the free energy.
And so I assume in this thing, what I've taken is every sequence is equally likely. Okay. And I ask myself, but with every fold is not equally likely, right? Because some folds have high free energy. In fact, the reason why this one, which has no bonds, is the most likely is because it has a high entropy state, right? So it's a no bond state. Right? This is done at room temperature. Right? So the argument is basically some structures are very favorable to free energy, and so there's many ways you can design that structure. Some structures are really hard to make. There's only like one or two ways you could design them. Does that make sense? That's basically the argument here. Basically, it's like in some places called designability. How designable is the structure? And this is designability of these things. And I'm saying to you, is I can tell you how designable they are just based on this random argument.
Sorry.
Yeah.
Are you fitting A and B or?
Yeah, so in this case, the dashed line is with no fits to A or B, and the black one is with fits to A and B. But you can pick all you need is one or two data points. You catch them, and then
But the A and B comes from your compression algorithm.
the A and B, yeah, well, they're linked to the compression algorithm. So the point is here, so this is interesting, right? I tell you that some things are exponentially more likely to appear than others, right? And then the question is, does that impact nature, right? Is this what right? Does it impact nature? So there's a longer story here, many little subtleties for people that are really interested in RNA. But the one thing I will do is that you can find somewhat simplified descriptions of these structures. Okay, so here's this is a ribo switch. It's called. I can find, and bioinformaticians have worked on. You can intuitively think, well, probably if I made this ring one big or one small, it wouldn't change that much. So you want to have some kind of coarse-grained description of it. The eventual coarse-grained descriptions that I have a smaller number of structures, so I'll find the same structure more often. And what's kind of interesting here is I'm going to show you two systems, 55 length and 126. That's the first hyper-astronomical number, the number of which the possible number of if I made every sequence way more than the mass of the observable universe. Here I've got the probability or the frequency with which these things appear on random sampling. And I colored all the ones that are found in nature. So these are the ones that we think are found in animals or plants or prokaryotes, basically. And what you see is actually I only find about 68 of the 10 to the 7 shapes for length 55, and only 63 out of 10 of a trillion shapes for length 126. Okay. So that means that nature is only using an extremely small fraction of that space of possibility. That space of possibility is enormous. Nature is using a very extremely small fraction of them. And I can plot this to you in a different way for length 55. So here I've done the first 183 out of 10 million possible structures at this level of coarse graining. And all the yellow ones are the ones that are found in nature. And I can basically tell you for the first of 44, every single one that I predict is found. Here's one that's not found. And as you go down, it gets a little bit more rare. But the point is, I can tell you what structure you're going to find in nature based on this coding theorem argument, and based on this probability that they appear. And this is effectively, not a survival of the fittest, but an arrival of the fittest argument, right? So these are the ones that arrive. Nature only works with that which arrives. It can't work with things that don't arrive. If things arrive with hugely different probabilities, then it's going to work with a small subset of all the possibilities. And so the argument is here we are giving a non-adaptive, so a non-fitness-based argument for why you see the structures that you do. Right? Each individual structure obviously has some fitness that's used for in nature. That's why it's conserved. But the probability that they appear, you can predict on this random ground. Go ahead.
So your first item on the list is just the boring dots or the brackets.
Yeah. So at this level of coarse graining, so those ones do appear in nature. Yeah, and they are functional.
They're functional.
Some subset of them are functional. They're tagged as functional in various databases.
That's actually number 43 here, right? the simplest.
Yeah, actually, I take that back. Yeah, be a bit careful. This one in this coarse grading technique, right? It's not quite the same as the one I showed you before, right? So the a bunch of them are in it's basically like a bucket of a whole bunch of different structures. And I think if I remember right, this one is not. It's not just the unbound one.
Because if you look at these pictures you showed, these data sets and
Yeah.
there just looks like that the boring ones or the not so useful ones are just orders of magnitude more likely than the useful ones.
Well, no, so actually, so I should say, I didn't put the slides in this on that. I have a paper just come out on bioarchive that looks exactly at this question and basically points out why you don't see very many of the unuseful ones. You see the useful ones. Actually, I now understand. I remember I gave you the wrong answer for number 43. So the point is these are buckets with a bunch of different structures that look similar to each other. So 43 is indeed the unfolded structure, but it's only one structure, whereas this one is a bunch of different structures that come into there. And so when you add them all up, the ordering is different, right? But there is only one unbounded structure, and that happens to be here, right? So it does appear in nature, but not the highest probability. So there's a little bit of a sleight of hand here that happened, which is that by using a biophysically motivated coarse grading technique, you bias a little bit of how you're looking at the structure. If you look just at the raw structures, then what you'll see, if you look at the probability of K for RNA, you see something that looks like this, okay? Which is that some of them are, that the low ones are very rare, the high ones are rare, and in the middle is the vast majority of them. And that's when our paper we've just written called RNA secondary structures are conserved but random.
I know nothing about proteins, but I'm wondering if we had like these sort of observations and data sets in time, if there is a relation between like that distribution of your K and when the new ones appear. Yeah, so one of the really interesting questions would be if you were to start from scratch, right? Would you populate this, slowly this direction? And one of our predictions is that as we keep finding more and more structures in nature, we might start populating these ones down here because they're more rare. Look at evolution history. Another thing I've slipped under the rug entirely, and it's a long, complicated story, is that evolution doesn't work by randomly picking sequences, right? Evolution picks a particular sequence and mutates around that, right? So there's an assumption here, which is true for RNA, which is that the frequency that local mutations happen is very close to the frequency of their global probability, which is the one I've put down here, right? But of course, what this doesn't include is that RNAs also change length. Okay, so in order to say what order these things appear, you've got to have a much more careful model of the evolution dynamics with time. Like global pressure plus local variation.
Something like that, exactly, yeah. I'll give you some other examples of that where it's a bit easier to understand, and you'll see but that principle is definitely there. All right. Yeah.
I have a question. So you said this experiment, they found all the time the same thing. Do you mean in this RNA in vitro equals? Do you mean they found the same structure or the same sequence?
Same structure. Yeah, not the same sequence. They found different sequences, yeah, but same structure. So that's the interesting thing, right? The phenotype, people talk about in evolution about genotype, which is the sequence typically, and the phenotype, or the thing that encodes the information, the phenotype, which is the biological organism. In this case, they keep finding the same phenotype. This is interesting if you're interested in convergent evolution. So the idea that same thing evolves multiple times, which we see all over the natural world. And the question is, I got interested in convergent evolution with my friend Simon Conway Morris, and then realized that this is one example I might be able to understand. Let me give you another example that's kind of maybe also interesting. So this is a bacterium. Bacteria swim with these rotors behind them that turn. This is a little video from Keiji Namba's group in Osaka of the self-assembly of the bacterial flagellar motor. This is a really cool video. In fact, it's this video is what got me interested in self-assembly on its own. Right? So what happens is your body makes these proteins, or the cell, sorry, the bacteria makes these proteins, and they spontaneously self-assemble into this really well-defined three-dimensional structure. It's an amazing piece of nanomachinery. it can rotate at 100,000 RPM, it can stop in a quarter of a turn. I have a colleague in Oxford, Richard Berry, who measures the torque of these things. Completely amazing. What's also amazing is that they self-assemble. So there's also interesting physics about how do you get a bunch of things, shake them around and form this well-formed motor. It's a bit of taking Lego blocks, putting some glue on there, shaking them in a box, and out comes a fully formed train. So the physics of that is very interesting. But there's another interesting question, which is how did evolution find this particular structure? It does so actually by having the individual proteins evolve patches that then make them stick to each other. Okay, so this is even crazier. It's like I take Lego blocks and I randomly change the way they stick, and I shake, and out comes a fully formed train. That would be even, if I could do that, that would be really cool. But nature does it all the time. So it turns out half the proteins in your body, in the protein database, are complexes. So they're dimers, trimers, tetramers, etc. And so here I see some protein complexes. You can think about these as little graphs, which basically tell you who binds to whom, right? And so we came up with a simplified model we call polyomino. So here's a little octamer. You can think about this in 2D as little squares that can stick to each other. And if they stick, if you get the right set of patches on the sides, like that set of rules, they should always come into this direction, into this shape. So that's a model we call polyominoes. It's just trying to model very crudely how these proteins assemble. So the evolution of these proteins is that they typically are first made not sticking to something, and then they evolve patches and they make these complex, including these machines. So here's an example of polyominoes, little blocks with, here I'm going to give the rules of little letters here. In this case, A binds to C. So if I simplify my dynamics by putting this number 1 down first, then if these things float around, the only one that's going to stick to is number 3, which has C. 3 has D's on the side, which stick to B. The one with B on is number 2. 2's will stick there. So there you see 3 stick there, 2's on the side. 2 little O's that don't stick to anything. So this set of rules will deterministically make this structure no matter what order I put them down. That's modeling in a very crude way the self-assembly of these structures. I can then ask myself the design problem, which is if I give you the structure, can you find a set of rules that would make it? In particular, can you find maybe the simplest set of rules, etc.? So it's a fun kind of problem to do. And my student, Ian, who's now at the University of Bergen, did this for 16mers. Right? So it's all possible 16mers. So polyominoes are a branch of recreational mathematics, and so people worked out lots of things about them, including that there are 13,079,255 different 16mer shapes. So he ran an evolutionary algorithm, which basically said, make a 16mer, but doesn't care which one. Any 16mer will do. Okay, and stop when you find a 16mer. So you evolve it and you mutate it. You might think you'll find any sequence, any structure with a probability of 1 in 13 million. But what he found in his runs is that half of the time he found this small subset of structures. Okay, and if you look at this more detail, you can look up a complexity argument as how hard is it to make this particular one? How much information do I need in my encoding to make this one? Then you find that the probability versus complexity follows this very nice 2 to the minus k rule. What's also interesting is I can look at these structures and say there's something special about them. So I've colored in dark blue the 5 structures that have D4 symmetry. It's the symmetry of a square. I can rotate them and flip them and they're the same. These are the only 5 in the entire set of 13 million, okay? And they all appear with very high probability. Why is that? Well, a symmetric structure doesn't need much information to describe it, right? If I want to tile this floor here, if I just say, take this tile and repeat it n times, that's a short description. If I tell you where every single tile is, that's a long description. If I'm randomly evolving things, I'm much more likely to find a short description that will tile the floor than a long description that will tile the floor. And therefore, I'm more likely to find a symmetric structure than an asymmetric structure. So symmetry and algorithmic information complexity are linked to each other in the sense that symmetric structures typically have short descriptions. Does that make sense? And that's why they evolve more frequently. So with the collaborator in Cambridge, Sebastian and Ard, we looked at the whole protein database and showed that basically if you use these same complexity measures and assume that something similar happened for protein assembly, you can explain the frequency with which you see symmetric structures in the protein database by this argument. So simple things are much more likely to appear than complex things because simple things are just easier to evolve. And this is a non-adaptive argument for why you see so much symmetry in the protein database. Symmetry may have other functional adaptive advantages, okay, but you don't need them to explain why they're there. So I'll skip that. This thing got a lot of information, a lot of symmetry, got a lot of. So I was going to write a paper about information theory and algorithms. And Ian said, no, we should write about symmetry. Yeah, and we did, and it got picked up by the news people, a lot of stories about it, which is nice. So we've done a lot of work on this evolution problem. I gave you two examples, RNA and protein complexes. I like them because I can actually understand what they do. I can actually have some stab at looking what the possibility space looks like. And I've shown you that that possibility space is hugely biased in terms of if I just randomly pick stories, which is randomly pick sequences, randomly pick books, randomly pick sequences, I'm exponentially more likely to get simple stories than complex stories, So sequences like the books, stories is like the phenotypes. I pick sequences at random, I'm much more likely to get simple phenotypes. And we've shown this works for gene regulatory networks. So these are how genes talk to each other. You can make predictions, and natural gene regulatory networks are typically ones that are very simple. Richard Dawkins has a very beautiful little model of development called biomorphs, which if you've read his books, you'll have seen. We show that those have a very high bias towards simplicity. So although he used them to demonstrate the power of natural selection, you can also use them to show the power of the variable variation. Another paper we just submitted, so the bioRxiv, looks at leaf shapes. And there we did some phylogeny, so evolutionary histories of leaf shapes, and showed that evolution is much more likely to evolve towards a simple leaf shape than towards a complex leaf shape. And we are able to predict the frequencies with which we find simple and complex leaf shapes based on a gene regulatory network model for leaf developments. And the last thing we looked at are teeth evolution. So these are seal teeth, okay? And what's really interesting about tooth evolution is that their natural selection is very strong, okay? Basically, if you get a more complex tooth and you're a herbivore particularly, then you can eat a new plant and you'll outcompete your neighbors. And so it's interesting because this is an example where what we see is there's an intrinsic bias towards simplicity. And what we see here is that, for example, in seals, the common ancestor of all seals, all pinnipeds, wide range of similar animals, did eat plants and have much more complex teeth than current seals, who have now just are pierce feeders. They just eat, they grab something and eat it. They need to have sharp teeth, and that's basically it. And so we're saying there's an intrinsic, here they're losing their complexity because natural selection is not working anymore. So this is why I'm interested in tooth evolution. I'm also interested in tooth evolution because there's been a generation of very clever biologists who've worked out these models that I can then use. Right. The last thing I want to talk about is how this applies to learning. Okay? Because if you think about it, there's some similarities between evolution and learning, and you can think about both of them in this algorithmic language. So I'm going to talk about deep neural networks, or DNNs that we sometimes call them. I'm going to give you a really simple example of one of the fundamental problems in the field. So here I give you maybe 10 data points, and I ask you to fit, say, a polynomial to it. Well, we teach our students, don't fit too big a polynomial. Here's this dashed line is a 20-order polynomial. You'll exactly fit your data, but you might predict very badly. In fact, there are many, many different 20-order polynomials that would fit that data, and that gives what we call a variance problem, right? So small differences in my fitting procedure give me completely different predictions for out-of-sample data. That's called variance problems. So we tell our students, pick a simpler polynomial. If you have 10 data points, maybe pick a 5-order polynomial. That's this dashed line. It can't quite fit the data, okay, but it seems intuitively predict better in between the data points. What we think that it's true that the real underlying system could be generating this 20-order polynomial, but we think it's more likely on intuitive grounds that this red dashed line is better. Right? And this is called a bias problem, right? So you can't quite fit the data, so you've got bias and variance. Now here I show you this green line, 3 different neural networks. They give you this line that goes like that. Interestingly, these are millions of parameters. Okay, neural networks, you can prove all kinds of approximation theorems. You basically say that any function that satisfies some very basic, it's the best integral, they can approximate as arbitrarily well. So a neural network could, on this data, fit that data with a 20-order polynomial or any polynomial you can imagine, right? Or any other function that could fit the data. It can fit all of them. And all you're training it is to give 0 training error. So why does it pick this green line, right? Rather than in one of the infinite number of other functions it could have picked? Right? We're not telling it that. We're just training on the data. Okay, something intrinsically is doing that. So that's the question that I want to answers. So from the possibility space of function, which is unbelievably large, why has it picked this very small subset that seemed to be quite smooth and not so wild? I'm going to give you a simple version of this, a model problem, which is learning Boolean functions. And I'm going to give you n is 7 Boolean function. So let's say that back in the dark days of COVID, you were a doctor here in one of the hospitals in Amsterdam. There's pressure on your hospital beds. So a patient comes to you, shows you a bunch of symptoms, is old, has some comorbidities, and you might send them to the hospital. Okay, maybe the same patient comes to you, they're young, but they show all the comorbidities. You might send them to the hospital. Maybe the patient clearly has COVID, but otherwise is healthy. You might say, sorry, sick at home, right? That's you, and etc. Right? So this is one doctor. Now, how many possible doctors are there? Right? There's 7 questions. 7 yes, no, that's 2 to the 7, 128 possible answers. Okay. So for each of the 128 possible answers, the doctor can send you to the hospital or not. So there's 2 to the 128 possible doctors. Right? So even with a small problem, there are 10 to the 38 possible doctors. Okay. Each of them is a Boolean function, which basically looks at binary inputs and gives you a yes or a no answer, 1 or 0 answer. So what does a neural network do on this data? Okay, you can ask a problem not too dissimilar to the problem I asked with the evolution problem, which is if I take a neural network like this, which takes inputs, which are the answers, and then has weights, and then gives an output, which is 1 or 0. I just pick these weights at random, which is like a monkey typing on a typewriter, or like me picking genotypes at random. How likely will that neural network produce one of these 10 to the 38 functions? Now, you might think it'll produce them all with a probability of 1 in 10 to the minus 38, but it doesn't. It picks certain functions with high probability. In fact, 10% of the time, it gives you all 0s or all 1s. It basically sends everybody to the hospital or nobody to the hospital if it was a doctor. And then these are the other ones that it finds. This is the complexity of the doctor. So I order the doctor's answers from first all 0s answers of the patient to all 1s of the patient, and then I can zip that and it gives me an idea of the complexity of the doctor. So simple doctors are exponentially more likely to appear than complex doctors when I start, before I've even trained, right? So I'm much more like, I'm biased. I have a very big, strong prior for simple doctors. Does that make sense? So the question is, what does this tell us about neural networks? Well, it tells us that they're biased towards simplicity, right? Already when you start, right? So if I start and I've got many, and I'm over parameterized, so I've got many possible solutions I could fit to my data, I'm going to exponentially more likely fit simple than complex ones. Now, can I break that simplicity? It turns out that there are some tricks you can do with neural networks. If you change their activation functions a little bit, they can hit a chaotic regime where you can prove in the full chaotic regime, they will genuinely predict every function with a probability of 1 over the number of functions. So if you make it totally chaotic, this one will predict every Boolean function with equal probability. Okay. So what happens is if I make them deeper or I increase the chaos, this is Occam, it kind of disappears away. Right? The reason this is interesting is because I can now have a handle. I can turn my neural network from a normal neural network into a chaotic neural network and ask myself, how do I learn problems now? Right? And the learning problem I'm going to do is you have a doctor, I give you half of the answers of that doctor, okay? And then you predict what the doctor would do on other sets of patient answers. That's a very standard problem. So first thing to ask yourself again is, I just gave you this, so just remind you, if I look at these possible doctors, right? The vast majority of doctors are complex, right? There's a small number of simple doctors, right? And just this question about compression techniques, a perfect compressor gives you this 2 to the k thing. My Lempel-Ziv approximator to complexity does pretty well, but then it kind of flattens out a little bit at higher ones, but I'm not going to care too much about that. So if I take that, that's the probability that if I take all possible functions, I'm exponentially more likely to pick a complex function than a simple function, because many more complex functions than simple functions. Now, if I give you half of the data points, half the answers, okay, that's 64 answers, I can still fit that data with any possible answer on the data I haven't seen yet. So there's 2 to the 64, 10 to 19 possible functions that neural network could fit if I give it 64 data points. So the question is, why does it work at all, right? Given that it could fit almost anything to the new data. So here I've got my prior. This blue is the way it normally runs, and if I make it chaotic, it slowly becomes less biased towards simplicity. This is a 2 to the minus k probability that I get an individual function of complexity of an individual function, right? This is the probability of all functions of probability k. So very few simple functions and very many complex functions. If I multiply these two in a neural network, a normal neural network does something really interesting. The 2 to the minus k exactly cancels the 2 to the plus k growth of possible functions. And you're almost equally likely to get a simple function as a complex function. So it kind of uniformly in complexity space finds simple versus complex. So these ones are, the density states is very large here. There's many possible functions, but you've got a small probability of getting them. These ones, there's very few of them, but you've got a high probability of getting them, and they kind of flatten out, okay? If I make my 2 to the minus k slightly less than 2 to the minus k, 2 to the minus 0.95k, for example, right? Then I no longer can kill those very large not density of states. And I see that my density states over in complexity space is hugely dominated towards complex functions. Does that make sense? So that's called the variance problem, right? The variance problem I told you about before. There are many, many complex functions. I'm biased towards them. So what does that tell me? If I now look at how well I learn, I gave you that problem where I give you half the doctors, 64 of the 128 so half the doctor's answers, right? So I pick a doctor, okay? And maybe my doctor is very simple. My doctor is everybody to the hospital, nobody to the hospital. I give you 64 of those answers, and I say predict the other 64. So neural network trains on those 64 answers I've given you and then predicts on the ones it hasn't seen, right? So my neural network does remarkably well. It basically does pretty well in these simple doctors. My chaotic one doesn't do very well, right? It typically picks more complex doctors than there are, even though I give it all zeros or all ones, because the number of possible complex functions that have all zeros and then something complex is extremely large, right? There are very few that are all simple and then stay simple. Up here is a random learner that literally just picks bits at random. And so the probability of you getting it right is 50% average. And that's a bad learner. But a small variation in my simplicity bias. I'm still hugely biased towards simplicity, but not biased fully, but by instead of 2 to the minus k, I'm like 2 to the my 0.8k, minus 0.8k. Already I'm lost. I can't predict very well, right? So here you see this doctor predicts really well on simple functions, but does worse and worse on more complex ones. Okay. What's also interesting is that there's a theorem called the no free lunch theorem that tells you that if I don't know anything about my system, no algorithm will predict better than random chance on average. So I know that my neural network can't do better than random chance on all possible Boolean functions, right? But the vast majority of them are here, right? It can do slightly less than random chance, and it'll all average out to basically no better, no worse. So why do neural networks work so well on predicting things? They only actually work well in this case on a tiny subset of possible Boolean functions, which are the simple Boolean functions. They don't do well at all on the vast majority of them. Right? So that's a basic principle of neural networks. They predict really well on simple things. They predict badly on all possible functions. And so the really interesting question is, why does this inductive bias work so well in the natural world? So why do you take your neural network, feed it some data, and predict it, and it does so well, right? Because given that data, it could actually predict mathematically a wide variety of other outputs, but it predicts something with this inductive bias, of which simplicity is one of them. I think there's many more inductive biases, okay? And those biases somehow reflect something of the natural world. So the very deep question is, why do neural networks somehow reflect something profound about the natural world? From this incredible number of functions that they could fit to their data, why do they fit data functions that then predict well on unseen data? It must be that they're somehow recapitulating something about the properties of the world. And so the idea, which I can't really I don't really know how to grasp, but I can at least point you towards, is perhaps it's because many of the things that it's trying to learn are somehow also being generated by a similar kind of algorithmic process. And that's why they match so well with the natural world. And so they're telling us something very deep about the way the world works. And this is how algorithms, something about simplicity or maybe other inductive biases as well, are somehow emerging from the algorithmic nature of the world. And when we use certain kinds of algorithms to learn them, and these match, they're telling us something about both sides of that equation. So I want to thank you for your attention. This is the group of my very talented students and collaborators who worked on the evolution side. This is the group that worked on the machine learning side. They did all the work. I go and talk about it and take your questions. And thank you very much for your attention.
So how do you know that for anything in the world that I measure, images, text, the space of things that I actually see is much smaller than the space of possibilities. So that's a pattern dimensionality.
Yeah.
Versus a generated rule dimensionality. So how do you know which one it is? Or
Yeah,
are they related?
Yeah, so I think that's a really deep question. So image is a nice example, right? So
Yeah.
if I look at all possible images, right? That's actually all possible sets of pixels. The vast majority of those are just noise, right? Only a very small subset of those images are actual things that we think are real live images. So they are a very small subset of the natural world. What's kind of surprising is that neural networks are very good at image recognition. They can look at images and they pick something up about them. So that's a map from pixels to labels, right? Which they do very well, and we do very well, right? And so that tells, but actually it's not hard to take a neural network and make it be very bad at learning those labels, where it still gets zero training error, right? But you can trick it in various ways and it doesn't do it well at all. So it tells you that the neural networks have some kind of inductive bias that is really good at looking at images and recognizing them. And the deep question is that, first of all, is the algorithm that it's using the same algorithm that we use when we, with our vision and our cognitive capacities, add labels to data, or is it using a different algorithm? It's not always so clear. So sometimes in early days, it turned out that when dogs and cats is one of the classic problems, that neural networks could actually do pretty good at dogs and cats by looking at inside and outside. Okay, so it's doing a relatively good job on labels, but not in the way that we are. Right? We now think that what they're doing is closer to what we would do with visual perception, but we don't. It's also a very deep problem because if I take a neural network and it's a very, very deep problem for society, if I take a neural network and use it to make some decision, I would like to know what it's making that decision on, right? If I think it's really good at recognizing dogs and cats, but it's really just recognizing inside and outside, right? Then I could get it. It could be very wrong in really important situations, in mission critical situations, or in situations that are bad for society. And so understanding what these neural networks are doing, they're still black boxes to a large extent, is super important. So that question there's no simple answer to that question. If I had the answer to that question, I would have my own startup telling everybody making many billions, right? And so it's a very deep and profound question. I'm just pointing out there's something magical about them in the sense that they are reflecting something about the natural worlds, but what it is not so clear. And so in the case of the image recognition is a nice example because we understand a lot about how animals and like ourselves process images, right? And so we think, in fact, some of the best image recognition neural networks are inspired by human. CNNs are basically inspired by the way the retina works. So there is some overlap with that, but I think there's a lot of difficult questions that have to do with the structure of data that we don't yet have the right tools to properly understand. Yes, and yeah.
So going back to the cellular automatons, if your doctor problem, this is going one shot, so you get the variables, you give an output.
Yeah.
There's no feedback into the system.
Well, there's feedback in the training algorithm.
Yeah, but there's no the output of the decision is not the input for the next decision. But if you take a cellular automaton, you quite easily map this to like a Langton 1DCA, where you can vary any of the variables. And that becomes a different problem to learn.
Yeah.
Because you've got to learn the rules and you've got to really learn the algorithmic process. And maybe that's something fundamentally different about the way that neural network would learn. I know some people at Google who are looking at these neural cellular automata now, trying to fit these.
Yeah, so I did one kind of supervised learning here where you pick a fixed set of data that trains zero training error, but you can also do all kinds of continual learning in this same problem where I keep giving you more data, right, and telling you the answer. So here I trained, what happens, I trained for 64 of the answers at once, right? Yeah. And then I asked you what to do on the other 64. I can also give you first the first one, then the second one, then the third one, the fourth one, but I still don't know what the rest of them are, right? So it turns out that it's more or less the same.
So when your prediction also feeds into your next round.
Yes, but the point is you're predicting. So if I tell you if I keep it's effectively it's a different training protocol for what are effectively very similar kinds of problems.
Yeah, what I'm trying to get is this idea of the computational reproducibility for some of the cellular automata, right? So
yeah,
whether these things are just fundamentally different in the way they're learned.
how the cellular automaton learn.
The class 4,
Yeah.
the complex ones. There's this idea of the computational reducibility. So the only way to kind of predict
Yeah.
future state is to simulate it.
Yeah.
Whether they can learn this, because there's probably a different type of learning to what's happening.
So that's an interesting question, right? So I don't know whether they would learn. I mean,
if you're doing physics-informed neural networks, the question is: are they learning the equations or just learning the times?
yeah, it's a super deep question about which there's lots of when I think a large language model, is it reasoning or has it just memorized a set of patterns, right? These are really deep questions. Generate, yeah, exactly. Has it learned exactly? That's a really good question.
One of the questions you
I mean, one thing I would say is basically there are some very simple generators like pseudo-random number generators that it can't learn. Right? So it has the things that can learn.
The idea of this universal computer, these all 30 things, and it becomes there is something happening, right?
Yeah.
Not just simply
Yeah.
One question just quickly about your symmetry. I noticed most of them have rotational symmetry as well, but you somehow distinguish the ones we.
I picked just the default ones have all those symmetries, the high symmetry ones.
Okay.
You can go down in symmetry classes. And roughly speaking, what you use, the vast majority will have no symmetry.
Yeah.
Okay? But so a very small fraction of high symmetry so actually I'm working.
It's just the redundancy, right? You can create that up there.
Many different ways, yeah, exactly. And so symmetry and this algorithmic complexity are correlated, but not perfectly. Yeah.
Yeah, beautiful work. I noticed that you focus very much on the complexity. Of course, that's part of the simplicity argument. But of course, if you read the literature also and listen to other people talking about machine learning or about finding solutions to DNA structure or RNA structures, I mean, the natural language is that of statistical mechanics.
Yeah.
So you do have minimization problems. You have here some annealing, for example.
Yeah.
So and I was wondering, you didn't mention that at all.
I didn't exactly.
Because this framework now relates to the framework of like annealing and diffusion results.
Yeah. So I'll take a step back and give you an answer to a simpler question than the one that you're asking, which is a more complex question. Right? So it's true that I used simplified language here. Statistical mechanics, you would say, in this design space, we're looking at entropies, right? So simple things have large entropies, large volumes, complex things have small designable entropies. Okay. What I skipped over because it's a longer story is in this plot, right? I tell you the prior probability you get a certain function if I randomly pick parameters. Okay. In this plot, I tell you the probability that I get a particular solution when I've trained using stochastic gradient dynamics, which is an annealing-like thing, right? And so what I'm claiming is that these two are working hand in hand. So what I'm actually saying, I'm saying is here I've trained with SGD and I see that simple functions I can predict really well, complex functions I can't predict well. And I'm saying that's linked to the fact that simple functions are really easy. Simple functions are much more likely to find than complex functions. And the way to think about that very briefly is to think about this in terms of basins of attraction. So if I have some kind of it can't even work. But point is, I have a basis of attraction. Okay, so I've got some kind of energy here, and then I've got some generalized parameter here. If I have, oh gosh, I can't really draw it. I got one that works. Good. So if I have something that looks like this, okay. The point is, these things are highly overparameterized, so they have many possible solutions to that to the problem at hand. When I train, I just go down a free energy, in this case, a loss landscape minima. There are many minima that are equivalent to each other. They're all zero. So if the minimum is large, I'm much more likely to fall into it, right? So if it's large here, it's going to be large there, and these ones are small, and that's the argument for why the size of these loss minima that the simple ones have large basins of traction, the complex have small basins of traction, I fall into these rather than those. That's the argument. There's a lot of argument in the literature about what causes the inductive bias of a neural network. And there's a very large community of people that come from optimization, a lot of mathematicians, who basically think it's the actual algorithm itself that does that.
It means optimization.
So some property of stochastic gradient dynamics does that. I am not in that. I am partially in that camp and I think it does do something, but I think that the first order, you can explain a lot of inductive bias based on this intrinsic bias. It's just there. I say that. So the reason, if you're using neural networks, right, what you know is if you change your learning rate or you change all kinds of little bells and whistles you can turn, it really improves your generalization. Right? And that is, I call that second order generalization. So it makes it slightly better. But the fundamental question is, given I gave you 64 answers, I actually picked out the 64. There's 2 to the 64, 10 to 19 possible solutions. Why do you pick, how can you ever get anything better than random chance? That answer is given by this implicit intrinsic bias story. Whether you can do slightly better than somebody else is a second order effect. That's what all you care about when you're a user. Right? Because you don't care about the big question of why is it working in the first place, because it works, right? You want to make it work slightly better than your competitors. So that is true. And then but not everybody in the community believes this. Maybe I'm wrong in some fundamental way, but I don't think so. Yes, go ahead.
Going back to the evolution story for a second, you gave some examples of seals and leaves where the genetic circuit is broken down into substance. So where the seal teeth, it's extending towards
simplicity.
Are there cases in evolution where it perhaps not present, but in the past where evolution has tended towards complexity, and if so, is there sort of like a trade-off between the simplicity of the output and evolutionary advantage, and how does that trade-off?
That's a really good question. So first thing I was going to say is, we're clearly complex evolution. So evolution doesn't just make us simple. And teeth are a nice thing to think about, because whether a complex tooth is advantageous or not depends on the environment that you're in. So herbivores, animals that eat leaves, basically have much more complex teeth than carnivores. And the reason for that is because every time you get a slightly more complex tooth, you're able to digest more. So there's a direct metabolic advantage that you get. And so that's an example of where complexity, if you're a herbivore, complexity is good and simplicity is bad. What you can show, and biologists have already shown this, if you take a tooth, a system, and you start randomly mutating it in an animal, the teeth are going to get simpler. So the evolutionary, the kind of mutation pressures towards simpler teeth, but natural selection is pushing the other way, and there's a balance between them that's working out, which is really nice. In the leaf shape example, we're able to explain the patterns, the phylogenetic patterns, really well in a model that ignores this natural selection side. And then the question is, why is that true? And I'm not really sure, but we're able to, we have a hypothesis that has no natural selection. We can explain why you see this kind of preference for simple leaves or complex leaves. That presumably means that natural selection is not hugely favoring the leaf shape, would be my thing. Now, there's a really deep question I think is hiding that you're also asking, which is, where does biological complexity come from? And I'm going to wave my hands, but I think one of the things that the bias simplicity does that I've not talked about is it also gives you solutions that are more robust. So, for example, in that self-assembly picture of those protein complexes, these simpler proteins, like the symmetric ones, also have the property that you can mutate their surfaces and they're less likely to break the structure. So if you want to make complexity in biology, the way you often works is modular. I have something that works, that does something, and then I add another module to it. So maybe I take 2 complexes and I put the 2 complexes together, rather than going directly from no complex to complex. And so that's always easy to do if you're more robust, because then if you're robust and you're 2 complexes, you can mutate another patch on your surface without breaking your initial function and become something more complex. And nature is full of these kinds of modular solutions. And so one of the ideas that I wave my hands about, but it's hard to be quantitative about, is that the way to get complexity is through these modular holes rather than directly from scratch.
You mentioned briefly about other complexity measures.
Yeah.
And in some of the plots, actually on the x-axis, you have different complexity
Yeah.
measures against the probability. Is it robust against any measure, or is it context dependent? How do you?
Yeah, so that's a very good question. So in these Boolean functions, okay, I gave you one complexity measure. There are actually, if you like the math of Boolean functions, there are all kinds of other complexity measures that you can calculate. And so one of the things that we always try to show is that these measures correlate with each other pretty well. And in the Boolean functions, they do. We think that for the Boolean functions, these more complicated complexity measures are better. So one example would be the parity function, right? Where you're basically looking at how many ones and zeros are there in something. Turns out to be a function neural networks have a very hard time learning, okay? But it's actually quite simple, okay? But Boolean complexity says it's complex, right? And so Boolean complexity is slightly better than this Lempel-Ziv complexity that we have. So we're working on that question. This is where the mathematicians, I think, rightly get a bit nervous because Kolmogorov complexity, which is where the theory comes from, is uncomputable. I'm using approximations of Kolmogorov complexity. I'm saying this scales like Kolmogorov complexity or similar Kolmogorov complexity, but it may not always be. And so this is why the Boolean functions are nice because they're foundations of modern theoretical computer science. There are lots understood about them, and I can compare these complexity measures. But that's a very deep and difficult question that I think we're still exploring. And maybe the last thing I'd say is we're pretty sure that lower in the Chomsky hierarchy, so things that are not universal Turing machines, that coding theorem-like theorems will not have Kolmogorov complexity in there, but some other measure. And we're working on what those measures exactly would look like.
I have a question. So in machine learning, you also have this idea of kind of information bottleneck or double descent of rocking that kind of first you go to a highly complex solution, and then there's kind of the inductive bias kicks in, and then you start to generalize well. So do you also have kind of an explanation for that in this story? Because I feel maybe that's different than the evolutionary story.
Yeah, so I am working on that problem, and I think there's two answers. You gave two examples that are slightly different. So one of them is double descent. Oh, God. One of them is double descent, right? So what is double descent? It says that the error that you make, right, with the number of parameters, okay, what happens is in your classic machine learning textbooks and still in classes today, everyone's talking about bias variance trade-off. It says you make your model too simple, you can't fit the data, right? You make your model too complex, and you get various problems. You find lots of solutions that are equivalent to each other. You don't know which one to pick, and you get big errors. And then what double descent shows that at some point, if you make your model more and more complex, which is what neural networks are, this goes down again. I think there's nothing deep about double descent. All it's telling you is that if you make your neural network really small, it follows standard machine learning techniques, stuff that VC dimension tells you, and that a whole generation of mathematicians and computer scientists have worked on. And this tells you that doesn't work there. Right? But we know that they work well, that they generalize well. So we know that it has to go back down. Right? And in fact, you can come up with more complex models that will have more than one spike. This is really well understood and can be understood in the framework that I just showed you, which is to say, on problems that you have this inductive bias, you're going to get this low error down here. The question of what people call grokking, which is sometimes one way of thinking about that, is I got a problem where I give it more and more data, and suddenly the neural network is able to fit that very well. That is actually usually, I think, linked to something called feature learning. So that's a longer story, but that's something which is beyond, it was actually precisely not picked up by this. So one of the, I'm trying to argue in a recent paper that the way of, so what that basically is, so maybe I'll take a step back and say, and so in 2012, when the famous AlexNet paper came out by Jeff Hinton and Ilya Sutskever and Alex Kurinsky, that kind of started the modern AI revolution. That was they had a competition on learning images, right? And one of the amazing things about neural networks was that they just took the images raw and didn't do any feature learning. So what happened before is if you were trying to do machine learning AI, you'd spend a lot of time looking at your images and picking out properties that you thought that you to train your model to learn on. These things just learned it directly. So the intuition has been that neural networks, their hidden layers are learning features. They're doing something like that. They're learning something about the data that simplifies it, and then they can figure out the end, what's a cat and what's a dog. We think that that feature learning is something that emerges by the dynamics of the neural network in various kinds of ways. And so that is something that's beyond this. It turns out for image data sets, that effect is not very large. So in other words, you can learn really well with what are called random features, where you just basically don't do any feature learning. But there are other data sets for which that is absolutely critical. And that's what we call, and so we think that grokking has to do with you learning these features at some point. Typical grokking experience. And so how important that feature learning is for the performance of like state-of-the-art neural networks is not clear, but it's probably very important.
For the talk, what do you expect is the dynamics in the sense that if I make a string of a time series of a solution of a dynamical system?
For what? For it? Dynamical system.
Complex relation with the survivor dynamic, like if I would get a time series or solution of a dynamical system, make a string, and then I can make the same probability complexity bounds.
Yeah.
Yeah.
So the claim that we make is if you have some system, I don't know what your system is, but some system is simple in some way or the other, and it has some kind of probability. So there has to be some, there's more inputs than outputs, right? You can get that maybe by coarse graining your outputs. Then you should see simplicity bias. Yeah. We've even shown simplicity bias for the logistic equation, which is probably the simplest dynamical equation. And so
if I get a brain time series, I would find it,
the prediction that we make is yes. Yeah. But with some caveats, I have to look at your system in more detail to tell you if it will work, but I think it should work.
Going back to this folding of these
RNAs.
Yeah, RNAs, these things. So we didn't discuss anything about folding energy or free energy and kind of things. So is it possible to sort of combine these things? Because if you find a very simple string that's very low complexity, but has a very high bending stiffness, you will probably not see it in nature, but it's not doing anything useful. So is it possible to sort of combine these things? You can sort of say, okay, we're looking for low complexity on low bending spaces that you sort of use this physics or chemistry informed.
So that's a good question. So so what's happening in the RNA case is that you give it a sequence, you put it through a free energy calculator, it calculates the free energy of all possible structures, and picks the lowest free energy structure, or maybe some Boltzmann distribution around the lowest free energy structure. So there, the language is the language of physics, and the algorithms are the sequences that you put into it. Right? And the claim is that makes simple ones versus complex ones. If you change the thermodynamic properties of that thing, it's going to make a slightly different set of structures. So what I can't tell you is what the structures are. Right? Your algorithm tells you what the structures are. But what I can tell you, no matter what you do, you can make the bending stiffness, you can just change it if you want to in the models, wouldn't be hard. You would run it with different bending stiffness, you'll get a different subset of structures. I'll still predict that the simple ones are exponentially more likely to agree with the complex ones. What I can't tell you from this very kind of null model is what structures nature will find. What's happening in practice is I got a particular structure, I look at all possible sequences, and I say this one is the free energy minimum. Well, that depends on the fine details of the system. So I would say maybe I'll say one other thing, which is, this is a grand theory, right? Like all grand theories, it's wrong. yeah. But hopefully not all the time.
Just a very quick final general question. What role does scale play in sort of describing things, in the sense that earlier you're putting these RNA patterns into different bins based on some kind of coarse graining?
Yeah.
And now in general, does it all kind of work the same way depending on what resolution your description is at, or does that?
Well, so the claim would be that it shouldn't depend on the resolution of your description. Yeah, I guess the principle should be the same. I use that resolution because there's a limited number of natural RNAs. So looking at a more coarse-grained level makes it more likely for me to find the same thing more than once in nature. So if I look at dot bracket notation, I never find the same dot bracket more than once, or very rarely. But at the more coarse-grained level, I do, and that allows me to calculate probabilities. But in principle, the scale is just I can scale afterwards how I like, and it should always work.